He's an independent plant and soil health educator, a healthy soils advocate and presenter on soil biology, plant nutrition and integrated approaches of sustainable farming. He's originally from Australia. He's now based in Canada, but has worked on both conventional and organic farms, improving biological farming practices in both Australia and the UK. And he consults with farmers all around the world, where he integrates soil, chemical and biological assessments along with plant nutritional analysis as a joined-up strategy for crop management. So I'm going to hand over to you now, Joel, for the next half hour or so to give us a presentation, and then we're going to have a Q&A. So do get your questions and comments in for him now. Joel.
Okay, thank you very much for having me. I'll just share my screen here. Okay, is that working? Hopefully, yeah. Okay, so thanks very much to everybody for having me today. I had the pleasure of tuning in a little bit yesterday and really enjoyed the content so far. So today we're going to talk on roots, not shoots, or building organic matter from below ground is going to be the focus. So let's dive straight into it. I've got probably quite a bit to get through. So let's start with images like this. You know, if we think about historically our productivity increases and yield increases, we've seen this steady trend over the last 100-odd years of you know, all sorts of science and technological improvements that we've used in order to increase yields over the last 100-odd years, and there's just a few examples of that. Now, of course, we might assume that based on the fact that yields and overall primary productivity has been going up in this time, therefore the amount of residues, crop residues, stubbles that are being left and integrated back into the soil should have also been going up, that actually our soil organic matter levels should have also been increasing over this last 100-odd years. But of course, with the intensification of agriculture, we've seen the exact opposite, where you know, generally across the board, soil organic matter levels have been declining with the intensification of agriculture. So it's a little counterintuitive there when yields have been going up. Now, the reason for that is that the vast majority of crop residues that does get integrated into the soil ultimately is oxidized off as CO2. Often as much as 75% of that residue is broken down and respired by the microorganisms and released as CO2. So we're only left with a very small fraction of the residues that do stay in the soil, that do get integrated into the soil organic matter. So these are the decaying plant compounds, the microorganisms that grow and feed on those, and some of those microbial metabolites, microbial byproducts, microbial exudates and things that ultimately make up a mix of what we might call soil organic matter. But really, at the end of the day, it's plants and their photosynthetic capacity, their photosynthetic potential, which draws the carbon in the first place through photosynthesis. It's plants that provide the carbon, but then we are dependent on the microbial interactions to then integrate that down into the soil. So that therefore means that we have three parts of plants, three components of plants to think about in terms of plant material that enters the soil. And so it can only be really either our shoots, our shoot litter, our root litter, or those plant root exudates. So these are the three fractions of carbon, of material that come from plants that microbes are exposed to, that they have to process, that they have to integrate down into the soil. So let's have a look at these three components then. Now, I've said that yields have been going up and productivity has been going up in the last 100 years, but if we look a little deeper, a little bit of the nuance in that, here we're looking at segregating out some of the different part components and changes that we see with modern varieties versus older varieties. And what we can see, a particular trend here, is that we're looking at root and a below ground biomass and above ground, and you can see that right around about the green revolution here, you can see that actually the amount of root biomass actually declined. Although we bred short varieties and high yielding varieties that overall increased our above ground biomass, increased yield, actually we saw that came at a cost. It was a trade-off, and we actually reduced the amount of root biomass that modern varieties grow. And this is another graph just showing the same thing. You can see right around there that the overall root biomass begins to decline, even though the above ground residues and the overall primary productivity increases. So when we say we've increased yields, really what we've increased is above ground biomass, and that has come at a cost to below ground biomass, to the root litter. And if we look at then the impacts of fertilization and fertilization intensity, what we're looking at here, this was a study on both wheat and corn, where we are seeing that as we increase the fertilization intensity, as we increase fertilization, that indeed that actually triggers greater biomass gains above ground. That is the effect of the nitrogen fertilization. If we look at high fertilized crops versus lower fertilized crops, actually their root biomass, the below ground biomass, is generally much the same. So the effects of nitrogen fertilization seems to increase again above ground biomass, not so much the below ground biomass. And this is another graph just summarizing that exact effect. You can see here on the bottom axis, as we increase the nitrogen fertilization, you can see that the amount of below ground allocation of carbon progressively declines. Okay, so we have two major factors there that have led to an increase of above ground biomass, breeding and fertilization, but that has, both of those things have come at a cost to the below ground biomass, to the root structures. Now that's really important because it is roots and not shoots that ultimately build soil organic matter. It is the decay, it is of the root litter, it is the below ground interactions, the below ground material that ultimately is processed by microbes and is stabilized into soil organic matter. So we have a very significant body of evidence that all points in this direction, that contribution of root litter is a much more important source than the shoot litter. So there's just a selection of studies there. I'm just going to cherry pick out one of these, the Jackson one here, because it's a nice review that looked at a lot of other studies, the ecology of soil carbon. And just if we quickly scan our eye down this table here, we're looking at comparing the below ground and above ground inputs and what percentage of those became integrated into the soil organic matter. So if we just very quickly scan our eye down this first column and say, well, what percentage of roots became soil organic matter? And you can see all of these numbers down here, 30, 60, 90, 30, 60, 80, etc. We can see all of these numbers down here, what percentage of roots became soil organic matter. And if we compare that to what percentage of shoots became soil organic matter, you can see that across the board, this second column, all of these numbers are much lower. Okay, so it is roots and not shoots that build soil organic matter. And the summary kind of point from this study, they suggest that those root inputs are approximately 5 times more likely than an equivalent mass of above ground litter to be stabilized as soil organic matter. Okay, so if the goal is to build soil organic matter, and of course this is a hot topic in today's climate with issues like climate change hot on the agenda, if the goal is to build soil organic matter, we have to be shifting our thinking to below ground. Here's a nice image illustrating this. We're looking at comparing, this is just a simple little pot trial, comparing an ancient traditional wheat on the left, an einkorn, versus a modern durum on the right. Okay, so here you can see the effect of that breeding that I talked about earlier, where we can see much more overall root biomass down here on the older, more traditional variety of wheat. And similarly, another photo illustrating a similar effect here. We're kind of comparing, for example, perennial plants. Of course, perennials, being the fact that they live year after year, they do allocate a lot more carbon below ground. So they do grow much larger, much deeper root systems. versus our annual plants and annual crops. So, of course, this transition from perennial-based systems, perennial-based ecosystems, into annual-based agroecosystems, that has, of course, been a major factor that overall has lowered the production of roots, of root biomass, and the amount of those residues that enter the soil, ultimately enter the soil and potentially form soil organic matter. Okay, so that's your take-home message. It is roots, not shoots, and therefore that means we really need to be putting roots at the top of our agenda in terms of focusing on those below ground interactions, how to grow more roots and grow deeper roots and then leave them there. And that is the key to building more soil organic matter, roots and not shoots. But what about then root exudates? They are another fraction, this third piece of the puzzle, shoots, roots, and exudates, that also can be integrated down into the soil. And what impact do these exudates have? Now, plants can exude quite significant amounts of carbon as root exudates. It can be 20, 30 odd % of their total carbon that they breathe in, they can release. For annual plants, and for perennial plants, it can be much higher, 30 up to 50% of the total carbon that they breathe in can be exuded out as root exudates. So they are a significant contribution of carbon, and it turns out that they also have been a neglected piece of this puzzle, and that they also do make an important contribution to building soil organic matter. And let's explore why that is. And here's a study that was looking at this exact question. They basically said, okay, well, if roots are more important than shoots, what about these exudates? And here, this study, as it says here, is providing evidence for the primacy or the importance of living root inputs, of root exudates and rhizo deposits, not root or shoot litter, in forming soil organic carbon. So I'll just read a few key points here. Recent theories suggest that those living roots inputs exert a disproportionate influence on soil organic carbon formation, but few field studies have explicitly tested this by separately tracking living root or root exudates versus the litter inputs as they move through the soil food web and into distinct carbon pools. We show that those living root inputs are 2 to 13 times more efficient than litter inputs in forming both slow cycling mineral associated organic matter as well as fast cycling particulate organic carbon. I'm going to come back to those two terms in a second, but the take-home message here is that they're suggesting here that those root exudates are a much more efficient pathway to build soil organic matter versus the litter inputs. So let's have a little look at why and how that is. And I've got a very technical diagram here to explain this one. So here we're looking at a theoretical little microorganism, bacteria, let's say. And that bacteria is going to ingest some food sources from the surrounding environment. And that bacteria, this microorganism, will take in these smaller components of carbon, these things that are more easily available, easily digestible. These smaller compounds are more easily assimilated by the microorganism. So they will feed on the carbon, they'll break things down into smaller components, and they will feed on these smaller components, and they will ingest those. Once they've ingested those smaller components, then they will use their various metabolic pathways and these what's called intracellular enzymes to then build their bodies. They will restitch the building blocks together, and they will build more complex compounds from the simple building blocks. These are the small, simple building blocks, and they will then build their bodies, build microbial biomass, build more complex structures from those smaller molecules, from those smaller building blocks. So the point is to say that if there was a more complex material out there in the soil environment, the microbes can't necessarily just directly ingest that as is. It's too large, it's too complex. So, for example, this could be something like lignin or cellulose. These are structurally complex compounds that the microbes can't just integrate into their body. They have to break them down first into smaller components, where then these smaller components can then be more easily assimilated into their body. So that means in order for our biology here to break this down, to feed on these complex compounds, they have to produce what's called extracellular enzymes, exoenzymes, enzymes that are on the outside of their cells. So they exude, they excrete these enzymes. These enzymes then attack the complex molecules. They will chemically cut and sever and break apart these complex structures. So it's a chemical process, enzymatic digestion, that then breaks these complex things down into the smaller components in which the microbe can then ingest. And so the point here is to say that these exoenzymes come at a cost. They come at a metabolic and an energetic cost. The microorganism has to synthesize these exoenzymes. It has to release those. And that energy expended, that metabolic cost of doing that, means that the microorganism doesn't have as much energy to grow more microbial biomass. So if I could say the microbe has to waste energy on producing those exoenzymes in order to get its food. If we delivered more simple molecules to the microorganism, it wouldn't have to produce as much of those exoenzymes. It could just feed on the more labile, the more available forms of carbon. It could just feed and therefore more efficiently grow more microbial biomass. It doesn't have to waste that energy. It can just feed more and grow more, put on more weight, so to speak. So the point then is to say it comes down to something called carbon use efficiency. These smaller things, smaller carbon compounds, are more easily digestible. They have a greater carbon use efficiency. The microbes can feed on them more effectively, more efficiently, and ultimately grow more microbial biomass. And so just kind of summarizing visually, this is kind of what I'm referring to, is that if a microorganism is given a choice, or it's provided with two very different food sources, one of them, rutexates, for example, highly available, highly easily digestible, easy to assimilate, versus some stubble, some residue, very high, very wide carbon to nitrogen ratio material, high in lignin, high in cellulose, very complex to digest. Again, so the microbe has to expend so much energy to break this down, to ultimately feed on it, to ultimately grow microbial biomass. So the root exudates have a greater carbon use efficiency. And this point about how easily or how efficiently we can grow microbial biomass becomes a very important point that we will move on and tease out now. So here then we're looking at the old view of buildings, the old kind of framework or the old thinking, the previous paradigm on how best to build soil organic matter. Previously, we thought that those root exudates could not build soil organic matter because they are too labile, they're too available, and the bacteria will rapidly digest them. They will release a lot of CO2 as respiration. And so we previously thought that root exudates did not make a major contribution to building soil organic matter because they were too small, too available, too fast cycled, and ultimately too rapidly respired off as CO2. So we therefore placed greater emphasis on the stubbles, on the residues, on the lignin type material, that more complex plant structural material. And we said, okay, fungi are the organisms who are better at feeding on that more complex material. And that is the stuff, because it's more complex, that takes a longer time to break down, that will be therefore more stable in the soil, have a longer residence time in the soil, that ultimately it is that litter that feeds fungi that then helps to decay and to build and break that down and to build soil organic matter. So we had this strong focus on residue, stubble retention, keeping stubbles at the surface, minimizing tillage, protecting, don't cut and bale stubbles, don't remove them. Keep that stubble there, because it is that stubble that is so important to build soil organic matter. That was our previous kind of thinking. Now, when we move forward, we're starting to understand how important that the root exudates and how important the microorganisms are. We have a new paradigm emerging that says that perhaps that this is not the most efficient way to build soil organic matter. And the reason for that is that we now are beginning to understand that soil organic matter, that a significant proportion of it comes from microorganisms. It's microbial bodies, microbial dead bodies, and then microbial metabolites, microbial enzymes and exudates and things that they release. That it is the microbes who are, and their microbial interactions, who make such a key role in building soil organic matter. So here's a study here that's highlighting that microbial biomass is a significant source. So plants still are the driver. They photosynthesize, they bring carbon into the soil. Bacteria and microbes, they will grow, they will feed on that carbon. But when they begin to starve, when they begin to die off, when they don't have any more food source, they begin to die. And as they begin to die, and then their dead bodies begin to break down, it is certain fragments in the microbial bodies, in their cell walls, that then integrate with soil, with the soil surfaces, and it is then these decaying bodies, these microbial necromass, dead microbial bodies, and their cell wall components that then chemically mix and integrate with the soil mineral surfaces and form more stable soil organic matter. So we're now realizing that this microbial pathway to building soil organic matter is much more important than we thought. And so here's a more recent study here that is quantifying this effect. And you can see here quantitative assessment of microbial necromass. So I'm referring to dead microbial biomass here, dead microbes, and their contribution to soil organic matter. We show that microbial necromass can make up more than half of soil organic carbon. Hence, we suggest that the next generation of field management requires promoting microbial biomass formation and necromass preservation in order to maintain healthy soils, ecosystems, and climates. So we're highlighting that dead microbial bodies are making up more than 50% of our overall soil organic matter. Even though it all comes from plants, ultimately it's the microbial processing and integration and transformation of that carbon that actually builds the more stable soil organic matter. And as illustrated over here in the graph, over here you can see that there's a suggestion here that it is the fungal necromass, the dead fungal bodies, that perhaps play a slightly more important role than the bacterial necromass. So it's particularly the fungal component having perhaps a more important role, but nonetheless, they're both very important. So then this basically summarizes this point to say that plant litter, material that's coming into the soil, be that shoots, be that roots, be that exudates, it all has to break down in one way or another and be processed by those, either by the microbes and consumed by the microbes, or, and that would be called an in vivo process, it goes internally into the microorganism because they feed on it, versus some of those enzymes that just externally attack the soil, the material. They're breaking it down ex vivo, like outside of the microbial cell, outside of the microbial bodies. And this creates two distinct pathways. When plant residues and exudates, when they enter the soil microbial body, when we grow microbial biomass, well, of course, that is the key, as we now know, to forming that microbial necromass, which is going to make up half of the soil organic matter. If that plant residue and litter never enters a microbial body, if it just gets broken down into smaller components through enzymatic attack, then that doesn't enter the microbe. So that fraction of the residues forms a different soil organic matter, which is not as stable. So the point then just here to say is that this was the study that was looking at this, highlighting, they call it the microbial carbon pump, highlighting that when plant residues, when it enters microbial bodies, that is the key to forming more stable soil organic matter. We want as much of our plant residues to go through a microbial processing, internal processing, as much as possible. That's the pathway that will lead to more stable soil organic formation. Now, it's not to detract from the ex vivo process, from the external breakdown. That can still be a valuable contribution. I'm going to come back to this in a second. It's not to say that that's somehow bad or worse, but just to highlight that they are different. Okay, carbon will either enter a microbial body or not. And if it's simple and available and easily digestible, easily assimilatable, then it's more likely to enter the microorganism body. grow biomass, ultimately become necromass and build soil organic matter. So this then comes back to our new thinking. Here's that old pathway again, where we discounted the role of root exudates because they were too labile, too available. They would be oxidized off as CO2 too quickly. So we focused heavily on litter, stubbles, residues. The new thinking now says that, well, actually, these root exudates and these more labile forms of carbon, they can play a very important contribution to soil organic matter, because these root exudates have a greater carbon use efficiency. We can grow more microbial biomass with more simpler food sources, i.e. root exudates. So the more bacterial biomass we grow, the more fungal biomass we grow, the more opportunity we have to then form that necromass and ultimately stable soil organic matter. So indeed, fungi, yes, they can break down stubbles, they can break down litter, but if you give the fungi the choice, if you give it some highly available, easy to digest root exudates, fungi will happily feed on root exudates if they get given some. They will also degrade the complex stuff if they have to, and they will. But if given the choice, they will more rapidly digest exudates. So indeed, exudates can build microbial biomass, and that can be preserved into stable soil organic matter ultimately as well. So this is the microbial pathway. This is the emerging paradigm that says, indeed, those microbial interactions going through microbial bodies, that is a central aspect to this. And therefore, maintaining living roots and root exudates to provide those root exudates becomes a very important focus. So this is part of the new paradigm or the new thinking in soil organic matter. These are the two terms that maybe you will come across more and more in future years. Here, then we're looking at those two fractions that I was kind of talking about. One that maybe does not enter a microbial body and just is broken down through enzymatic decay, external attack, versus one that does enter the microbial body. So what we're seeing here is that when we have low quality litter, so stubbles, residues, high carbon to nitrogen ratio material, this type of residue ultimately forms what is called particulate organic matter. And that's because it is not very digestible. It is not very easily assimilatable to the microbes. So it's more of the enzymatic attack pathway, and it forms what's called, therefore, particulate organic matter. Now, that particulate organic matter has it's more fast cycling and has a lower shelf life, a lower residence time in the soil. It will only live in the soil for 1 to 50 years. So it's not quite as stable. Now, that differs when we talk about higher quality litter residues, so things like legume residues. So here we're talking about a low carbon to nitrogen ratio. So legume litter, legume residues, and things like root exudates, these more easily assimilatable fractions of plant material. These higher quality litters, they, of course, can enter the microbial bodies. They can be digested by microbial bodies more efficiently. So we're growing microbial biomass. And when we grow microbial biomass more efficiently, as I've just mentioned, then we can form those dead bodies and form stable soil organic matter. But these dead microbial bodies, they have to be bound to soil mineral surfaces. So this is called mineral-associated organic matter. This is where the dead microbial bodies get stuck to and stick on the soil mineral surfaces. So that means that sandy soils, lighter sandy soils that have lower soil mineral content, of course, these soils have lower potential to form mineral-associated organic matter. Loamy soils, heavier soils, clay soils with lots of mineral surfaces, they can form lots of this mineral-associated organic matter. And it's this mineral-associated organic matter that is much more stable. Okay, it has a lifetime of 10 up to 1,000 years. Okay, so to summarize this key point, better quality litters, i.e. legume litter, lower carbon to nitrogen ratio, or root exudates, this plant material will be more likely to feed microorganisms more effectively. When those dead microorganisms get stuck on mineral surfaces, this forms the very stable fraction of soil organic matter, which we now call mineral-associated organic matter. So then this came up a little bit with Christine's lecture yesterday, when we talk, therefore, then about that soils can get saturated, that soils have a top end, they have a limitation on how much soil organic matter carbon they can sequester. This is only true when we're talking about mineral-associated organic matter. So we're comparing the two here. This is mineral-associated organic matter here versus particulate organic matter. And you can see that the mineral-associated organic matter, yes, it plateaus off. We can't keep sequestering carbon because we've saturated all of the soil mineral surfaces. And once we've saturated the soil mineral surfaces, it plateaus. And so again, of course, a light sandy soil will reach that plateau at a very low level. A clay soil will reach that plateau much higher. So that is fair to say. However, when we talk about particulate organic matter, although it's not as good quality, although it's not as stable as the mineral stuff, as the mineral fraction, as you can see here on the graph on the right, it does not saturate. We're not reaching a plateau. It's not dependent on mineral surfaces to be stabilized in the soil. You can see the upward trend there. It seems to continually sequester more carbon. So that becomes the fraction of particulate organic matter. Although it's not as stable, it still is a very important fraction of soil organic matter for this reason. We can keep building it. It doesn't saturate like the mineral associated organic matter does. Now, therefore, what is then key to preserving this particulate organic matter is aggregation. We have to trap that lower quality organic matter. We have to trap it within aggregates. We have to embed it within good soil aggregates. Of course, this aggregate here is a ball, it's a sphere, it's a 3D structure, and therefore all of the organic matter embedded within that ball or embedded within that sphere is protected when we maintain aggregate stability. As soon as we cultivate the soil and that aggregate breaks apart, that is the key where we will then begin to lose that particulate soil organic matter. We will be breaking down the organic carbon that is embedded within those aggregates. So preserving aggregation and maintaining that aggregate stability in soils is absolutely key to keeping soil organic matter in there. So again, topics of cultivation, we want to try and minimize as much as possible. No recreational tillage, for example. I broadly agree with Christine's comment yesterday. I think a little bit of disturbance is fine. I'm not a hardcore zero tiller. I think there's a lot of environmental context dependency there. I think in dry climates, being zero till is really, really important, and stubble retention is super important. But I do think small amounts of disturbance is also, I think, okay. But the key message is still very important. We have to be minimizing soil disturbance. We've got to preserve these aggregates to trap that soil organic matter and carbon in there. So that's the kind of a nice summary slide here. We want to drive plant productivity. We want to drive photosynthesis, drive, yes, root, shoot, and exudate production. That will then, those exudates particularly will help to drive the microbial growth. They can grow their biomass, then they can die, they can produce their necromass. Those necromass fragments can then be attached to either mineral surfaces or embedded within those soil aggregates, where they will be preserved as that mineral-associated organic matter, or the particulate organic matter will be held within those aggregates. And so if we look at root exudation and some of how do we enhance this process, how do we feed microorganisms as best possible to grow microbial biomass? Well, here we're comparing crops, annual crops versus grassland in terms of the carbon input by the roots. We're looking at those rhizodeposits, those root exudates. And you can see on the pink dots here that it is our annual plants that they have a peak of root exudation around about 50 odd days. So the bulk of root exudation will happen in the first 4 to 5 to 6 weeks, first 50, 60 odd days. The root exudates will peak at that point in time in an annual plant, and then it will decline and drop right off. So now maximum root exudation has happened, and it then steadily declines in an annual plant. If we compare that to grassland and perennial plants, well, you can see that they will also have a peak of root exudation in that first maybe 6 to 8 weeks, a little bit longer. But then you can see with a perennial plant that the decline of that root exudate is much more gradual. So, of course, this is when the message is we need to grow more perennials. This is why we see better building of soil organic matter in perennial-based systems, in grassland systems. It's because, one, we grow more roots, and two, we have more sustained production of root exudates as well. Okay, so the shift to perennials is a major strategy if we to build soil organic matter because they grow more roots and more root exudates. And so, of course, that comes down to things like grazing management. I won't dwell on this. It'll be discussed enough through the course of the 5 days. But the point being is, when we overgraze, when we graze too hard, that has a negative impact on root function, on root biomass, and also that root exudation. So trying to minimize the amount of grazing, taking around about that 50% grazing strategy, helps to then preserve root growth, preserve root activity, preventing those short-term complete stoppages of root growth and biomass. What about the effect of animals? Of course, animals being highly vilified lately. Here we're looking at a study highlighting that grazing enhances the below-ground carbon allocation, biomass, soil carbon, and this is an example from Florida, so it's in a subtropical setting. But still, there are other studies that illustrate these same trends. But this is a really interesting one because basically they took a pasture and they split it in half. And on one half, they put animals in, and on the other half, they excluded the animals. And they just cut and mowed and slashed the pasture versus the animals grazing. So where they excluded the animals, where there was no livestock interaction, they found that grazing exclusion, no animals, was associated with less below-ground carbon allocation, less root biomass, less root exudation, therefore less microbial biomass, and then overall lower soil organic carbon. Okay, so when we keep animals away, when we keep animals off the pastures, they don't produce as much overall soil organic matter. We need animals in there to also be part of that system. One more quick slide on the effects of plant diversity. So what about a diversity of root systems to build soil organic carbon? And here we're looking at a global meta-analysis. I'll just read 2 quick points here. By analyzing 1,001 paired observations of plant mixtures and corresponding monocultures from 121 publications, we show that both soil organic carbon content and carbon stocks are on average 5 to 8% higher in species mixtures than in monocultures. These positive mixture effects increase over time and are more pronounced in deeper soils. These species mixture effects are consistent across forests, grassland, and cropland systems and are independent of background climates. Okay, so across all of those different climatic conditions and production types and systems and ecosystems, across all of that variability, the trend is still very clear, is that greater diversity of root systems is also a piece of the puzzle. It does also help to contribute to higher soil carbon sequestration, more soil organic matter below ground. So I think, the take-home message then is, roots, not shoots. And I would encourage people, you've got to be looking below ground. Take your spade and you've got to be assessing your crops, assessing your pastures, assessing your cover crops by looking below ground. Do not judge their merits or judge their success by simply what you see above ground. And here's a good example of that. This is a photo from Scotland. You can see this is a cover crop. You can see on the left, it's a pretty small cover crop. It's not a big showy amount of biomass above ground. You can see a bit of bare soil here, and here's another photo. These 3 photos are the same field, same day. Here's another close-up. We can see a bit of bare soil. We only have a few inches of biomass. However, below ground, I look at this last photo on the right, and I think there is great value to this cover crop. There is still plenty of root biomass below the ground. The plants, they will grow down first before they grow up. So judge your success by what is going on below the ground. Now, of course, would I prefer to see this cover crop 2 or 3 times high? Of course I would. Of course, there are benefits to above ground biomass in terms of weed suppression, soil protection, etc. But so, of course, I think we can all agree we'd like to see this bigger and better. But nonetheless, it's not to say that this cover crop is bringing no benefit because it's so small. It has a wonderful root system below ground that you can see here on the right. So, in summary, then, roots and root exudates, they play a critical influence on influencing all sorts of soil properties, the soil chemistry, the soil physics, the soil biology. And as we now understand that they also play this very important role in building soil organic matter. It is that root litter that is more important than shoot litter. Okay, the goal is to grow more roots. That is your key. We have the emerging evidence, the new paradigm, the new thinking, the microbial pathway to building soil organic matter that suggests that perhaps those root exudates may be even more important still. Or even if not, they are still a very important and major contributor. So we still need to be thinking about both roots and exudates, both of those below ground pieces of the puzzle. But still, microbial transformation of these carbon inputs is key. We need the microbes there to process, to break down, to transform, to assimilate, to grow that microbial biomass, to produce that microbial necromass. But we also need to physically protect those dead bodies and that soil organic matter. Bind it up within aggregates is especially important, particularly in lighter sandy soils that have less mineral surfaces in them. So all of this then really suggests and points to this, all of this new thinking points to that perhaps the living roots soil health principle should be taking much more of a front seat in our management strategies. It is the living roots, keeping the living root and the root exudates turned on for as much time as possible throughout the entire growing season, all year long. Keeping living roots in the soil is clearly a very, very important strategy that perhaps should be much higher on our focus. And so how can we manage for more soil organic matter? How can we do, what are the practical takeaways? What are the practical strategies in which we can do this? With our cash crops, it could mean choosing plants, choosing crops that have a higher root to shoot ratio. So plants that have big root systems is simply what I'm saying. So that could be certain species selection for that. Lucerne, for example, nice big deep root system versus clover has a smaller shallow root system. Okay, just an example. We want big roots. Automatically, the transition to perennials is just an obvious win. As soon as we have more perennials, we have greater carbon allocation below ground. We have bigger root systems. So that's just an obvious one. But potentially, what about breeding new varieties, okay, for those below ground traits? There's definitely an opportunity to, if our modern varieties have been bred to have lower root biomass, we can also breed them to have greater root biomass. So I think moving forward, we can look at varieties that do have a better optimization of that root to shoot ratio. There are certain inputs potentially that we could use to help encourage more root biomass. Nutrition, for example, I mean, obvious things like phosphorus, sulfur, boron. These minerals are very important to grow root biomass. Okay, so optimizing nutrition is a piece of the puzzle, but also many biostimulants have a role here. All sorts of things can encourage more rooting activity. Seaweeds with particularly high in oxons, vermicast extracts. These things are all great biostimulant and root enhancing materials. Maintaining living root, that soil health principle, we can keep it simple. The use of cover crops or relay intercropping, keeping living roots in the 2 shoulder seasons beyond the main cash crop. Simple strategy, grow cover crops, avoid bare fallows, minimize disturbance, as I mentioned. Preserving that aggregate stability is really important there. The transition away from monocultures towards diversity, so the use of polycultures there. So we have a variety of root systems, root architectures. That's all going to help, as we touched on that big meta-analysis there at the end. maybe the use of companion crops or intercrops that could be chosen specifically for their rooting traits, specifically for their root biomass. Grow one cash crop for above ground harvest, grow another companion for below ground harvest, below ground root production. Cover crops, again, this will be next generation cover crops. Here we are playing around with cover crops. It's all very good. I think the next generation will be much more fine-tuned, looking at not just species of cover crops. Everyone's talking about different species and diverse blends and these kinds of things. The next wave will be choosing varieties, choosing varieties of cover crops that have better root to shoot ratios, varieties that will grow more root biomass and prioritize that over shoot biomass. And again, so the use of, say, legumes, which produce that better quality litters, so the lower carbon to nitrogen ratio litter is more likely to be digested by microbes, more likely to form that mineral-associated organic matter. And lastly, livestock integration. So using animals, I shared that study of grazing exclusion, livestock exclusion, using things like rotational or mob grazing where possible. All of this gives more time for more root recovery, more root activity, and it is ultimately roots, not shoots, that build soil organic matter. So however we can encourage more roots, ultimately the better. Okay, thank you.
Joel, thank you so much. That was just fascinating. So interesting to hear all the information there. I suppose, looking briefly at the ratio, I mean, if anyone is going out tomorrow to their land and digging up and having a look at the roots, can you give some indication of what would be a healthy ratio of roots to shoots, and what should they be looking for? I mean, you had a very instructive visual there as well.
Yeah, I think we often would say that plants generally they allocate roughly equal amounts of carbon to above and below ground as they grow. So it's about this kind of 50-50, but in reality, roots, they constantly grow and die, they grow and die, they grow and die, they're constantly turning over. And of course, then there's a lot of root exudates that get excreted out. So when you pull up a plant, very often the roots look a lot smaller than the shoots, because that part of that reason there's been this continual turnover and a lot of root exudates just excreting out. So, you might more likely see something like a 60-40, 70-30 kind of look visually to the eye. So I think if you can basically improve anywhere from that, really, I think if you could get it closer to visually trying to see equal amounts, if you could visually see kind of like a 50-50 balance in your biomass above and below, then you're definitely on the right track if you could see it to that extent. The other then key is that, again, as Christine talked about yesterday, and as many of you all will know, that you're looking for rhizo sheaths. You're looking for soil to stick to the root systems. You don't want to see white, clean roots. You want to see soil stuck to those roots. And so visually, if you're seeing that happening, I would say that's also an indicator that you've got good biological functioning there and the system is working for you.
And it's obvious thing to say, but it's the shoots that pay the bills. So I'm wondering, with these new varieties that you're talking about, the roots to shoots ratio, new varieties coming on, is it one at the detriment to the other, or do you think these new varieties in the future are going to provide both?
Yeah, that's right. Yeah, it is a trade-off. So that is why root biomass has declined in the last 50, 100 odd years. It's because we've got better at breeding for above ground. So yes, it is a trade-off. The more carbon we begin to allocate down to roots, that may indeed have negative impact on yield. Now, so I think that has to be, of course, factored in. And I think there's a lot of context dependency there. If we were, again, maybe a little bit different in Ireland, but you guys are a bit more temperate and have, the luxury of a bit better soil organic matter. But
yes.
Because of that. But if we were to take this into a dry climate, a hot and dry climate with very low soil organic matter soils, there could be an argument that would say, because these are hot, dry climates, they have often moisture as the biggest limitation. So there could be an argument that could say that if we were to spend a few years growing these higher rooting varieties, producing more roots, ultimately then building more soil organic matter, that may help to overcome the limitation, the moisture limitation. So you might see a short-term lowering of your yield to build soil organic matter, to build soil moisture holding, therefore build resilience. Then when you go back to your other varieties, you'll see better yields out of them because now the soil has greater resilience, greater moisture holding capacity. The other thing that must be balanced off here is that when you grow a bigger root system, you also have a bigger root system to scavenge more minerals from the soil.
Yes.
So you're going to need less inputs, you're going to need less fertilizers. So there will be a cost saving from growing a bigger root system, which can also help to offset the perhaps slightly lower yield.
Okay, that's very helpful and clear. A lot of questions, so I'm going to fly through them because we have about 6 minutes or so to get through this. Frank G asks, I'm in a very low rainfall conditions. Could the microbial population be competing with the crop for water?
Yeah, I guess that's theoretically possible. But at the same time, microbes and plants, they're so inextricably linked. They're almost inseparable. So in a way, we know that microbes, they need moisture too. So you're right in your thinking, but microbes can also help to supply moisture to plants. They can also help us, obviously the big example would be mycorrhizal fungi. They can tap into very nooks and tight spaces, nooks and crannies, and access moisture and deliver that to plants. But equally, plants can share moisture back to microbes too. So they're so inextricably linked that I would say that the two of them would find some kind of equilibrium, irrespective of your moisture content.
Okay, thank you. Another question, which actually struck me as well, from Therese O'Keefe. Is there a soil test to measure microbial biomass or to quantify otherwise?
Yes, there is. So this would be called microbial biomass carbon is one kind of common test, but you can do microscopy tests. So many of you be familiar with the soil food web approach. That's kind of what they're doing. They're using a microscope to measure the microbial biomass. So yes, if you were to, I'm not sure where the questioner is from, if she's from Ireland or not, but
I think she is.
If you were to look up the kind of the soil food web approach, then using a microscope technique is one way to measure microbial biomass. There's many other techniques in which we can measure microbes, more high-powered kind of genetic and molecular tools that we're also using more at the coalface of this. But yes, there are different ways to measure microbial biomass, and you can do it.
And just out of that, on that point, I mean, I've met a couple of farmers in my time who have microscopes on their farm, and that's exactly what they're doing. Is that something that farmers can do themselves, that they can learn to do, or is it something that you have to send it off to a lab?
Yeah, absolutely. No, with 1 day's training with a microscope course, there are many farmers around the world who do this, and they can do it themselves. It is a bit of a crude assessment. It's perhaps not as refined or precise as a laboratory test, but in a way, measuring anything microbiologically related is a little bit of a crude, blunt instrument. They're a vast and complex world, and we don't have any great tools to really study such complexity and such diversity. But that aside, yes, as an on-farm tool to make relative comparisons, to compare good and bad fields, yes, this is something farmers could do themselves with a microscope.
Super, thank you. They're a couple of £100, the microscopes. Nigel asks, if soils are waterlogged for weeks on end, is there anything we can do to help the microbes survive and flourish?
Maybe not so much in the short term, but the longer term would be to grow more roots, deeper roots, change your mix there, get deeper rooted species into there, get those deeper roots growing down, restructuring the subsoil so that you will also see better water percolation out the bottom of the soil. So longer term, I would grow more diverse root systems with deeper root systems. Yeah. Okay. You will, even under waterlogged conditions, you will have some microbes that will still function under those conditions. These are called facultative anaerobes. They can thrive quite happily under low oxygen conditions. So you'll have some that will still be functioning, but many will be dormant. But your longer term strategy is to grow more deeper roots.
Thank you. John A asks, does soil temperature matter? If nothing happens under 6 degrees Celsius at the microbial level, are we chasing a dream?
Yeah, I mean, microbial activity will decline at cold temperatures. They can go lower than 6. Many of the species, you're right in saying that, many of the species would have shut down and they would go dormant by that temperature, but you will have many extremophiles who actually can still function all the way down to 0. So you will have some cold adapted tolerant species who will still be functioning. But of course, yes, you're right to say their metabolic activity, their basal respiration, it will be very low. It will be very minimal functionality at that time. But if that's the case, that kind of means the whole system is dormant anyway. What you're then wanting to do is, you've just got to play the waiting game for temperature to become warmer, and then when things will function, you want to be ready for when things wake up. I wouldn't say, therefore, just because microbes go dormant, that this is a futile effort. You have to do what you can do during the on season, and you've got to play the waiting game during the off season.
Okay, and finally, there's a lot more questions coming in, but just time-wise, and then Sean has 2 questions for you as well. This is anonymous. Hi, Joel, big fan of your work. Do you think the less labour route of, say, Gabe Brown is a more long-term farming solution, or do you think that works in theory, but in practice may not be quite so productive?
Did you say less labour?
Less labour route, yes.
Yes. Does that mean labour or larva? I wonder. Okay, anyway, if you mean less labour as in move to perennials and just, use animals, move away from cropping, sure. In a way, you could say perennial systems are less labour. You keep a permanent cover, you don't have to re-establish it, use diesel fuel every year and prepare a seed bed, etc., etc. So I think from that point of view, yes, that's good. Then you will typically see better nutrient cycling with that transition to perennials and animals. I think that is a good system. I think that's one of the problems we have in modern agriculture today, the segregation and specialization path that we went. I think we do need reintegration. And I think that helps with, all of that nutrient cycling to minimize some of the problems of longer term soil infertility. So maybe in a right down in 100 plus years, you may run into some issues with fertility. But if you also grow deeper roots and start to scavenge into more of your subsoil mineral reserves and grow topsoil, again, the problem you speak of may be a real one, but it is a problem many hundreds of years away. I would say a much more urgent and pressing problem is excess fertilization, excess nutrient burden in the environment and ecosystems. These are priorities now that should be addressed immediately. The problem of maybe running out of fertility in another couple of hundred years is one for another time.
Okay, and as I said, Joel, we've had loads of questions, so you're obviously very much engaging everyone who's watching, but Sean has 2 questions for you.
Yeah, hi Joel. Joel, very quickly, ADAS in the UK identified some measures that would be appropriate for environmental schemes in the future, and they divided them into low impact and high impact systems. There was one that really I couldn't understand really, was they looked at growing cover crops, and grazing them with livestock, and they presented that as a low impact, whereas growing cover crops which are not grazed is determined to be a high impact. And I've just wondered, could you shed some light on that? Would you understand why?
When you say high impact, do you mean high benefit?
Yes, I think so.
More, so they're saying that by not grazing them is more beneficial?
Yes.
Mm-hmm. Okay. Yeah, I would disagree with that. Probably their argument is they're thinking that, like a lot of people do, that the shoots play a valuable role in building soil organic matter. They're thinking, let's keep residues, let's not cut and bale stubbles, let's not cut and bale covers, let's not graze them and remove them. let's keep shoots in the soil, and that's going to lead to building more soil organic matter. I think that's probably what they're thinking. As the evidence that I presented today in the lecture suggests, that roots make a very small and minor shoots make a very small and very minor contribution to building soil organic matter. So I think that the idea that we should preserve shoots and keep them in the soil, keep them in the system, isn't as important as we once thought. So I would argue that if you take those shoots, put them through an animal, that actually you open the opportunity to, one, make money, have an economic gain, but equally that grazing impact also can set the cover crops back to a vegetative stage and also turn the root exudates back on. So I think you will see greater root exudation if you see more grazed covers versus non-grazed. And as we also talked about, it's the root exudates that go down this microbial pathway to building soil organic matter. So my guess is they're just under that assumption there that shoots make a valuable contribution to soil organic matter, which they don't. It's not to say they're nothing or no importance, but they're a minor importance. I would disagree with that.
Okay, thanks, Joel. And one last question I have in from Trevor Harris. What is the role of larger soil biology from worms to arthropods and the nematodes in the breakdown of surface litter? And does this then add to the soil carbon?
Yes, absolutely. Very good question. And I had a very microbial focus here in the lecture today. So yes, you're right in raising this point. I would say that shoots can make a very important or a more important contribution to soil organic matter that they can do only when they are passed through, for example, an earthworm, through an animal digestion. When an earthworm comes up and feeds on those shoots, digests those shoots, takes them back down, then they have more potential to then go through that microbial pathway and be integrated. So I think that you do raise an important point. The insects, they burrow, they shred, they break it down, they mix the organic material. Earthworms, of course, actually digest it and then inoculate it with biology. Under those larger macroorganisms, and that does illustrate the importance of soil insects and arthropods and earthworms, that they then can help also improve the efficiency of building soil organic matter, particularly from the shoots. I think when shoots pass through an animal digestion, be that a livestock or be that through an earthworm, that is an appropriate way to use shoots. Roots should stay in the soil, but shoots, I think, should go through an animal or an earthworm, an insect. I think that then they can be much more valuable.
Joel, thank you so much.